Hi. Yeah, I guess it's running. Yes. Let's see if this works. Bismillah ar-Rahman ar-Rahim. So, I am Sayyid Abdul Wasi and I'm here to present on a specific part of the hallmarks of cancer. My hallmark is evading cell death. This includes the paper Hallmark of Cancer and Hallmark of Cancer's Next Generation, right? So, and other papers that I did research while I was looking into this. So, when it comes to cell death, the major thing that always comes into our mind is apoptosis, which is programmed cell death, right? Apoptosis is actually a type of cell death that is essential for the normal development and function of multicellular organisms. So, there are some major morphological steps of apoptosis, which is cell shrinkage, cell fragmentation, cytoskeleton collapses, nuclear envelope disassembles, and cell release apoptotic bodies. Right? So, this right here is a diagram of a flow chart of the signaling molecules involved in cell apoptosis. Now, during programmed cell death, you have two major pathways that you need to consider, which is one is extrinsic pathway and intrinsic pathway, which we'll be delving into in a while. But before we do that, I would like to draw your attention to a few key acronyms. BACS and BAC, which are pro-apoptotic genes, BCL2 and BCLX, which are anti-apoptotic, BH3 proteins, which are regulators of BACS and BCLs, IAP, which is an inhibitor of apoptosis protein, and FADD, which is fast associated death domain. Please keep these in mind. These are going to come in a bit, right? So first off, let's look at extrinsic pathway. The extrinsic pathway is pretty simple. The extrinsic pathway is when an exterior signal is sent to kill the cell, right? In this, the fast receptor binds with the fast cell ligand, right? And this activates the FADD, which we just talked about, the death domain, the fast associated death domain, which in turn activates or cleaves pro-caspase 8 and turns it into caspase 8. And then caspase 8 goes along and activates caspase 3. Then caspase 3 causes a cascade of caspase activation, which in turn eventually results in the death substrate being activated. With the death substrate being activated, apoptosis can proceed, right? This is the extrinsic pathway. Now, let's talk about one second. I'm having a little bit of trouble positioning myself. Sorry about that technical difficulty. All right, now let's talk about the intrinsic pathway. It's a little more deeper than that. Intrinsic pathway is usually when there is some sort of DNA damage. This could be through radiation, chemotherapy, or whatnot. The DNA damage results in a P53 response. The P53 response then starts to upregulate BACs and BAC, which are, as we said, pro-apoptotic. Now, when BACs and BAC come into play, what happens is that they essentially they reduce the permeability of the mitochondria, which releases cytochrome C, and cytochrome C goes and binds with APAF, which is apoptosis protease activating factor 1. Now, when APAF and cytochrome C are binded together, what happens is that they activate caspase 9. Caspase 9 then goes and activates caspase 3, right? Which then results in the death substrate being activated in the same apoptotic process, right? Now, what was the point of telling you all of this? The point is that now we're going to look at the mechanisms used by tumor cells to evade cell death in these processes. And how do they do that? They do this by, as you can see on the diagram, there are 6 points, right? The 6 points in the diagram. Those are where the points where tumor cells can come and actually mess up with the pathway a little bit and then cause apoptosis to not occur properly. So first from the top, they can start by reducing the CD95 level, which will in turn cause nothing to happen downstream right here on up top, right? So, secondly, what they can do is, as you can see, the second mark, the death-induced signaling complex. What they can do is inactivate the death thing. Sorry about that. They can inactivate the death-induced signaling complex by the use of lyse proteins. Now, thirdly, what they can do is they can reduce the emission of cytochrome C, well, the cytochrome C from mitochondria as a result of upregulating BCL2, because BCL2 is generally present, right? And it is anti-apoptotic protein or gene. So what happens in this scenario is that this is already present generally so that the cell doesn't kill itself all the time. So what happens is these guys, the tumor cells will upregulate this so that the mitochondria keeps the cytochrome C locked in, and when the cytochrome C is locked in, it won't come and bind with the APAF, and that won't result in caspase 9, and that won't result in caspase 3, which won't result in apoptosis. Now, what happens after that is that on number 4, they can actually reduce levels of pro-apoptotic BACs by loss of P53. If they can inhibit the P53 response, there is no BACs, which won't result in any mitochondrial any cytochrome C being put out, and so on. So after that, another thing that they can do is they can do the loss of APAF1. This can be done through the inhibitor. Sorry, this sorry, loss of APAF. Right. This is them just removing the apoptosis. Sorry, I forgot the. One second, my bad. Yes, this is the removal of apoptosis protease active apoptosis protease activating factor 1, right? So then on number 6, you have the upregulation of inhibitors of apoptosis, right? This is the one we just talked about, IAP. Over here, IAP at the end, very end, as you can see on number 6, at the very end, the apoptosis inhibiting factors will be inhibitors of apoptosis will be upregulated so that caspase they will combine with caspase 9, which won't result in the caspase 3 being activated, which won't result in apoptosis, right? This was apoptosis. Then the next generation, the paper, which was the hallmarks of cancer next generation, they actually suggested two other mechanisms that might have some involvement with cancer, which was autophagy. For starters, autophagy is a little confusing because it does have a positive and a negative role, so we're not sure whether autophagy is good for cancer cells or bad for cancer cells, like, I mean, good for us or bad for us in regards to cancer cells. Like, as you can see, there are conflicting thoughts over here too. Just wanted to add this as one. Now, for starters, what's the mechanism of autophagy? Autophagy is basically a process by which cells break down and actually recycle cellular organelles in response to cellular stress, such as nutrient deficiencies, right? Autophagy actually involves the formation and fusion of intracellular vesicles called autophagosomes with lysosomes, leading to the degradation of intracellular organelles. The autophagy process has both regulatory and effectory components, including proteins that mediate autophagosomes formation and delivery to lysosomes. So, the signaling pathway which is actually involved is PI3 kinase, AKT, and MTOR kinase can actually inhibit autophagy when survival signals are present, and downregulation of this pathway can actually lead to the induction of autophagy or apoptosis. Okay, so one more key thing to notice over here is that the autophagy has, like I said, two things. It can actually be tumor promoting and also tumor suppressing, right? we will delve a little more deeper into that over here. So, when it comes to the positive effects of autophagy, right, for us, which is that there was this research done which was called non-degradable autophagic vacuoles are indispensable for cell computation. Over here, what they did was they took mice, and in those mice, they impaired the RAS V12 transformed cells. Right, there was impaired RAS V12 transformed cells were surrounded by normal cells. All right, what happens is this reduced lysosomal activity, and reduced lysosomal activity unsettled autophagic activity. Now, when the autophagic activity is unsettled, non-degradable autophagosome facilitated apical elimination of RAS V12 cells. And what happens is when you didn't have a proper when you didn't have a proper autophagosomes, basically, what would happen is that cell there wasn't there was an inhibition of competition, which is when there was an inhibition of cell competition, it ended up causing chronic pancreatitis like lesions in mice, right? Sorry. Okay. Yeah. So, what are the negative effects now? So, what could actually happen is that autophagic degradation of the immune checkpoint proteins. The two proteins involved are PDL1, which generally enhances T cell activation and actually suppresses tumor growth. So, but what happens is in cancer cells, PDL1 and another checkpoint protein, CD47, I think we discussed about this a bit earlier, is both of them actually get encapsulated in endosomes to prevent their degradation. And another thing is MHC1 and 2 autophagy actually enables cancer cell immune escape and inhibits antigen presentation and T cell activation, right? So that was autophagy. Now, the third bad boy, which was necrosis. So necrosis is kind of similar to apoptosis, but not really. It's the polar opposite of apoptosis to some extent. Apoptosis actually ends up with cell shrinkage and then like fragmentation and gone. With necrosis, what happens is that the cell swells up and bursts open. In necrotic cell death, what happens is there's pro-inflammatory signals which are released into the surrounding tissue microenvironment. So, in necrosis, the cell is actually bloated and explodes, which releases its content into the surrounding tissues, right? Necrosis essentially for us has usually been viewed as a system wide exhaustion and breakdown, but now we are beginning to discover that, oh, it might actually be under genetic control in some circumstances. Necrosis actually releases pro inflammatory signals into the surrounding cells, unlike apoptosis and autophagy, they don't do that. So now the key takeaways over here are that necrotic cells can recruit inflammatory cells of the immune system, which can actively be tumor promoting by fostering angiogenesis, cancer cell proliferation, and invasiveness. Right now, necrotic cells can also release bioactive regulatory factors that can stimulate neighboring viable cells to proliferate, potentially facilitating neoplastic progression. Necrotic cell death may ultimately do more harm than good in the context of cancer and neoplasia, and potentially invasive and metastatic tumors may tolerate some degree of necrotic cell death in order to recruit tumor promoting inflammatory cells. Like, I mean, if they survive the necrosis, they can actually use that entire system to facilitate their own growth because there's pro-inflammatory signals that are involved in the pathway, right? So that is all from me. Thank you. I hope you enjoyed or learned something. Bye. Peace out. And how do I stop this is beyond me. All right. Thank you. Thank you. Bye bye.